the structure, yes, we've talked about this, but I can say I'm starting to review for the exam now. All of the bacteria, at least nearly all, there's those couple exceptions, have a cell wall, and in that cell wall there's going to be some amount of peptidoglycan. Okay, and then the key about the gram-positive is that they've got a really thick peptidoglycan layer, so thick that they need the teichoic and lipoteichoic acids in there to help stabilize. The analogy I used, the cinder blocks, the bricks, that is your peptidoglycan. The mortar between that holds it together are the teichoic and lipoteichoic acids. All right. So your gram-negatives, remember, will have a very thin layer of peptidoglycan, but they don't need the teichoic lipoteichoic acids. There's just not enough there. They don't need that stabilization. But they're going to have these other molecules. I won't mention them by name because yes, they're in the gram-negatives. They are not in the gram-positives, like the lipopolysaccharide or endotoxin, the outer membrane. So we'll come back to those next week when we spend 4 hours talking about gram-negatives. But just remember, you've got lots of peptidoglycan and those two lipid acids, but you don't have a couple other proteins and things that the gram-negatives have. All right? And you are going to need to become very familiar with these algorithms, because this is what, anytime we're thinking about identifying the bacteria, you're going to run through these algorithms. And when we talk about staffing in a couple minutes, I'll show you how you put this into practice. Several different versions. So here is one. There's going to be a second one on the other side with different levels of detail. But basically, you're going to start here with your gram stain, and then is it gram positive or is it gram negative? And you just keep working through that tree, through this algorithm, and you start to get all of that biochemical testing that we talked about last week. All right. This, for your exam questions, I can guarantee you, number one, because I wrote them yesterday and the day before, but you're going to have some questions that say a bacterium was isolated. Here's the biochemical fingerprint. Now tell me what the organism is, okay? Because that's exactly what you would be thinking about as a physician, as working in that bacteriology lab when you have an organism you don't know what it is, or maybe you're doing quality control at a biopharmaceutical company and you have to verify every so often that what you have is still what you think you have, okay? So there's one, or I guess you can say there's two versions there. There's a third version. Whatever works for you, but again, where you've got, instead of having all of the information here with the organism, like you did on the last slides, you've got it in this chart, okay? Whatever clicks for you, be thinking about it. And then this is kind of unusual. Usually there's the algorithms don't have quite this level of detail on the gram positive rods, and we're not going to, we'll hit some of the points, but a lot of the hardcore biochemistry is going to be on the gram-positive cocci, the staph and the strep. All right, so where do you start this when you start the process of identification? You're going to take that sample, whatever you get from the patient, the environmental surface, what your supervisor in that biotech lab hands you, and you're going to culture it, because you need a purified organism to do these biochemical tests. Oh yeah, so these big blue stars, sorry, I posted this New Year's Day and went down to see my parents at Christmas. They were sick, I came back and got sick. So yeah. The only thing these two, and it's actually on a couple of the slides, where I had supplemental slides, and then as I was finalizing the lecture, I didn't change the slide number. So I put these in as I go through the slides to say, hey, remember before you post it to update it, with the final version and change the slide number. I didn't. So that's the only thing those blue stars mean when you see them. So everything else is okay. It's just going to be this slide number. And in this case, yes, they're wrong. I took out several slides to try and trim the lecture, but yeah, there's a couple of these and it'll be a different supplemental slide, but that's the only thing that means. Everybody okay with that? All right. So you've got your culture. The next step, since we're talking about bacteria, is you're going to do a gram stain. All right. And since we're talking about today, what we're going to see is that we've got gram-positive organisms, because we're talking about gram-positives. And in this hour, we've got gram-positive cocci. All right. So now we're working through that algorithm. We've got gram-positive, we've got coccus, round. Remember, cocci are round, bacilli rods, okay? And then that's when you start doing all of your biochemical tests. But your very first thing, your very first pieces of information are going to be the gram pattern and the morphology from that gram stain, all right? And then you might be able to see how they're growing. Are they growing in clusters, or are they growing in pairs, or in chains? Now, bacteria isolated in culture, because they're all in that really tight, close-knit community, they're going to stick to each other very easily if that's in their nature. You will not necessarily see the very large, and I've got a good slide of that in one of the organisms we're going to talk about, but you're not going to see these large associations when you're looking at a patient sample, because it's growing in the body. You've got the blood flowing, you've got the sneezing, you've got the coughing, whatever, that helps minimize those connections that they're on that agar plate when they're just sitting there growing the bacteria, their normal association, okay? But since we are talking about in culture, you also have that piece of information. What's their community morphology, not just their individual morphology, okay? Then the very next thing you're going to do when you have a gram-positive coccus, you're going to do a catalase test. Remember, we talked about the catalase test. That's where you're looking for if the bacterium produced catalase, they can now inactivate the hydrogen peroxide of host defense. So yeah, it's a test we can use that really helps differentiate, but it's part of their virulence factors, all right? So remember that, yeah, we've got all these great tests, but that's not why the bacteria do that. It's not their name tag. That's part of their defense mechanism in some way. Okay. Good rule of thumb, not rule of thumb, this is hard and fast. If it's catalase positive, when we're talking about gram-positive cocci, if it's catalase positive, it's going to be some species of Staphylococci. If it's catalase negative, it's going to be strep. Some species, but it's going to be strep. Okay? Well, now we've got a lot of organisms eliminated one or the other way just based on a gram stain and one biochemical test. Does that make a little bit of sense? Okay. All right, we'll come back to that again here in a few minutes. Again, a little bit of a review, thinking about your antimicrobial therapies. The big one for gram-positive bacteria, there's two factors. One isn't really a therapy, but remember way back in, probably in July, if not really early August, in our host defense lecture, when I said, haha, in your tears and your saliva, one of your defense, barrier defense elements, the chemical defense, is lysosome or lysozyme. And I said, whoop, in micro, we'll talk about why that is. Here's that moment. Lysozyme specifically will cleave the cross-linking between your peptidoglycan. Remember your peptidoglycan, those repeating saccharide units of the GLAC-NAC, okay? Lysozyme specifically cleaves between those two, right? When you start cleaving between those two bonds, between those two disaccharide units, now your cell wall falls apart, your peptidoglycan falls apart. Neat, right? That's why it is. So that's a great defense mechanism. Now our beta-lactam antibodies, beta-lactams are a big class, so your cephalosporins, all of your psilins, your penicillin, amoxicillin, etc., all are beta-lactamases because one of the key structures in the molecule is a beta-lactam ring, all right? That is a very similar mechanism of action of your beta-lactamases, the penicillins and all those. They're going to cleave, they're going to inhibit, they're not going to cleave that bridge, but they're going to inhibit that transpeptidase. Because remember, you've got that tetrapeptide hanging down from your GLAC-NAC molecule, and now there's the transpeptidase is going to link those together. The beta-lactam antibodies prevent that transpeptidase bond being formed, right? And that's the mechanism of action of the drugs, right? So just remember that, and that's all you need to know about the drugs. We're not going to talk about them, but I just want to make sure that you understand broad spectrum antibiotics. So broad spectrum are ones that have a very wide area of activity. They're not going to be effective against just gram-positive or just gram-negative. So penicillin, really, really good, really highly effective against your gram-positive or any of your beta-lactams, but weak efficacy against your gram-negatives. Why would that be? Really thick peptidoglycan layer, really thin peptidoglycan layer. So it's going to take a whole lot more stuff to kill that gram-negative as far as a drug treatment, because they aren't as dependent on their structural integrity of that peptidoglycan. All right, but you can start with that penicillin. You can start with those beta-lactam drugs and have some efficacy, but then begin to target it if once you find out, I know this is a gram-negative, and now switch to something that would be more highly effective, more targeted. All right, so just because we use the term broad-spectrum antibiotics a lot, we won't do it a lot in this lecture, but just so that you know that term. All right. Okay, let's talk about staph. So the Staphylococcus genus, they're all gram-positive cocci, facultative anaerobes. Yes, why is that? Because some of them, a lot of them, are not growing on the surface of the skin. They're growing in deeper tissue where they need oxygen, but it doesn't have to be very high quantity. So that's why they're facultative anaerobes. That's how that Staph aureus is able to cause that boil, that carbuncle, able to cause that abscess, where there's going to be a very limited amount of oxygen. Okay? They grow, and this was the image I referred to earlier. So the Staphylococci, yes, coccus round, and they grow in these grape-like clusters. This is a purified culture. See all of these big clusters of grape? Everybody can see that? All right. Now, here is a patient sample. And you see, well, yeah, there's one really little cluster, but most of them are kind of, yeah, they're sticking together, but they're not in these really big clusters. So that's what I was referring to earlier about being able to differentiate. And just because I see this, I'm going to say, well, this can't be staph. I'm not seeing those big clusters. All right, I'm looking at in vivo or ex vivo. I'm looking at a tissue sample, something coming from the body where there's going to be all those forces. All right. Okay, so we've done our gram stain, we've got our coccus, we've got our cluster-like morphology. Next thing up, they're catalase positive. That's the next test you do. And here's that catalase test, where the bacteria are expressing catalase, so they're going to cleave that hydrogen peroxide. When you cleave it, you make water and you make oxygen, and that's the bubbling of that you see. The catalase negative, okay, well, since I don't show you the picture here, this would be if it were strep, catalase negative. There's going to be bacteria there. See how there's a little bit of cloudiness? So bacteria have been mixed in that solution containing the peroxide, but they don't have catalase, so they don't cleave it. So they just sit there. All right, that's your catalase test. And in the lecture last week, I think I had a link to a YouTube video, a minute and a half or so long, that shows you that. All right. Several different species of staph. The ones that are clinically important are these three: Staphylococcus aureus, Staph epidermidis, and Staph saprophyticus. And we're going to talk about each of those three in varying levels of detail. All right. So here is where we'll see how you kind of work through the algorithms that were on those first couple slides. All right. So if we're thinking that we have staph, or if we've got that sample and we're working through it, we say we've got that gram stain. It's a gram-positive coccus, and there it's purple. Remember, P positive, P purple. And now we've got a coccus, so we're going to go to this part of that algorithm. We've done our catalase test, and it's catalase positive. That means it has to be some species of strep. So now we're going to do the next test in our chain, and that's the coagulase test. And there was a link to that video, to a video in the last lecture as well. So the coagulase test is really important in two ways. For the staph which we will discuss, the only one that is coagulase positive is Staphylococcus aureus. All of the other staph are coagulase negative, and you will see that often abbreviated CO for coagulase negative Staphylococci. All right. So why is this important? In the body, that coagulase is going to cleave the fibrinogen to fibrin, and now that allows all the staph to stick together, all the Staph aureus to stick together more easily, to stick to that cell wall, to stick to that valve in your heart to cause endocarditis, to stick to that knee joint to give you septic arthritis, or that prosthetic joint if you've had the knee or the hip replacement to now give you that infection. All right, that just helps adherence, and so now the bacteria are held in place so they can grow a colony, cause that focal infection, but it's also now immunoevasive because they're basically putting on a coat of fibrin, and that helps escape the immune response because it's a host protein. Okay. What also helps, some virulence factors that all of the Staphylococci share are attachments, so they've got fibronectin binding protein, so that helps them to stick to that host tissue, and they make biofilm. Okay. And remember what the biofilm is. It's that protein carbohydrate shield cape that the bacteria make that they covers a community, and now it is well, not completely impervious, but it's difficult for antibiotics to penetrate, and as well as anything from the host immune response. Phagocytes can't get to them, cytokines can't get through that, complement can't get through that. All right. Skip to this bullet, but I'll go back. Remember, the mannitol salt plate that we talked about in the last time we were together. So this is how you, number one, can see if the staph will grow, because only Staphylococci will grow on the mannitol plates. But now also, and we've got a picture of this in a couple slides, only the Staph aureus will ferment the mannitol, and we'll see what that looks like in a couple minutes. All right, and then here's your coagulase test, where if it's coagulase positive, you get that clot. It's not truly a clot because you don't have activation of the clotting cascade, but you get that aggregation of the protein due to all of the fibrinogen and the plasma being cleaved and bound by the bacteria. All right, epidemiology. So Staphylococci are everywhere. A lot of them are living on us. Staph epidermidis is part of our microbiome on the skin. Staph aureus is not part of our microbiome, but lots of us, at least 30%, are colonized with Staph aureus in the nose, in the nares. Okay, can be colonized in other places. All right, Staph saprophyticus, as we'll see, colonizes the genitourinary tract. All right, and we'll see why that's important when we talk about the different diseases. If you look over here, and this is an image from our text, you can see that staph can cause disease at just about any site in the body. Now, most of that is Staph aureus, but not all of it. Okay, and we'll talk about some of the individual diseases. But it's, it can go anywhere. Transmission, Staph aureus, because it is everywhere. Just about any way you can think about a pathogen getting into your body, Staph aureus will do it. Okay, Staph epidermidis is part of our microbiome. So when it causes disease, that means that it's gotten into the wrong place at the wrong time, so it's an opportunistic infection. Right? You have that surgical process, and the surgeon isn't very good about cleaning, or himself or you, and so now Staph epi gets into that surgical incision, or it's on that prosthetic joint that gets implanted because it's we're touching things as we not if it's a very good production company, but you get that Staph epidermidis onto that prosthetic joint before it's placed inside the patient. Okay, and then Staph saprophyticus is also opportunistic because of it's colonizing the GU tract, and then it can get inside the body instead of just more on the outer surface, and you've got infections in certain people, and we'll see that in a couple minutes. All right, Staphylococcus aureus. So we've talked about some of the general virulence factors that all of the staph have and share with those attachment factors. The coagulase is also an additional attachment factor because of cleaving the fibrinogen. So these are the extra virulence factors that Staph aureus have. It's got a lot of other virulence factors that the Staph epi and Staph saprophyticus don't have. That's why it causes so much more disease. It's because it's got all these extra factors. Got lots of different enzymes. It's got some exotoxins, some of them that are really key in causing disease. Staphylococcal food poisoning. Why do you get food poisoning? Because entero, that's the GI. Anything enterocyte, enterotoxin, something that's happening in the gastrointestinal tract, right? So enterotoxin, these are the toxins that are made when you eat that potato salad that's set out at your Labor Day picnic with all of the mayonnaise in it, or the ice cream that was made with raw dairy and has Staph aureus in it. Lots of different ways. So Staph aureus, food poisoning, very often, not always, very often associated with dairy products. All right. Some other toxins that we'll talk about with a couple diseases. Toxic shock syndrome toxin number 1, so TSST1. That is a super antigen. All right. Exfoliative toxins A and B, so that's scalded skin syndrome. We'll see that in just a minute. All right. Then they have different proteins for immune evasion. So when we were talking about virulence factors last week, we talked about the IgA protease, an enzyme secreted by some of the bacteria that cleave that IgA molecule. All right, Staph aureus doesn't have that, but it interferes with antibodies in an even neater way. It expresses this protein called protein A. Okay? And what protein A does, it binds the Fc region of IgG with high affinity, IgM at a lower affinity, but it will still bind it. And so now it's basically lassoing that IgG molecule, anchoring it to the surface of that staph bacteria, but it's on the wrong end. It's tying up the Fc region, so complement can't bind. That molecule, that IgG, can't bind to an Fc receptor because the Fc receptor is not accessible. So now you've interfered with the antibody function, not just of anti-staph antibodies, but anti-anything antibodies. There's no specificity. Any IgG will bind to that protein A. Right? And then you've got the drug resistance, and with Staph aureus, we've got that whole MEK-A gene that gives us MRSA. All right, diseases. For Staphylococcus aureus, we're thinking about 2 different classes mainly. So purulent, inflammatory purulent, lots of pus. Right? What's the pus? We didn't talk about this in immunology, which I think we did. Pus is basically when you've got lots of neutrophils that are being recruited. That's pus. And it's degrading the tissue, it's degrading, if it's in this case an infection, and now you've got all of that yuck. That's the purulence. And then you've got toxic diseases. And so toxic diseases is when you have that toxin that's being made, and that's causing the inflammatory response. Might have a few neutrophils there, but really it's the toxic damage. You're not going to have that purulence, you're not going to have that pus. All right? Okay. So, maybe a little more detail here, but things to think about because, again, case-based questions. All right. So when we're thinking about food poisoning, so yeah, when we talk about the gram negatives next week, we'll go a little bit more into food poisoning. It's really many different things. But bottom line, somehow that food gets infected. Most often it is a human, and especially with Staph aureus, it's a human person, because remember, colonization, more than 30% colonized with Staph aureus in the nose. And so now, this time of year, sniffles. In the summer, allergies, sniffles. People wipe their nose, don't wash their hands, and then they go back to fixing whatever they're fixing, right? So, but somehow, or you're sneezing, or you're just shedding through respiratory secretions. But you're shedding, that Staph aureus gets into the food. Now the bacteria are going to grow in that food if it's not stored properly. Again, in the mayonnaise in that potato salad sets out at room temperature. Great, rich environment, nice temperature. The bacteria are going to grow. They're going to replicate. They're going to do their basic metabolism, which means secreting those exotoxins and secreting those enterotoxins that are heat stable. Okay, so that's why some of those toxins, heat stable means you can heat it, you can boil it, you can fry it, and it's not going, that toxin will not be denatured. It's heat stable. So now, even though you've cooked it, you can ingest it and you get sick. Yes, ma'am.
If you keep the food cold like you're supposed to, does the bacteria not get a chance to grow? Like, does it have to be left out?
For some of the bacteria like staph, yes. And I say that, I put that qualifier in there because we're going to talk about one at the end of the next hour that grows really well at 4 degrees. But as a general rule, yes, you keep it cold. Yeah, I've seen all sorts of faces. Yeah, if you keep it cold, so below about 10 degrees centigrade, the bacteria aren't going to grow, and if they can't grow, they're not going to metabolize, they're not making their toxin. Yeah. You're welcome. Okay, so food poisoning. what that you hear is a poisoning because you're eating the toxin. At the time that the food is set out to make enough toxin to make you sick, the bacteria have died. All right, so you're not actually infected, you're poisoned, you're intoxicated. All right, so whoops, wrong way. So because you aren't formally infected, no fever, not lots of those symptoms that you typically think of with a staph infection. All right, and I need to pick up the pace here. All right, toxic shock syndrome, most strong association here. Tampon use, women are infected, or not infected, they're colonized in the vaginal tract or in cervical tract with the Staph aureus. You use that tampon, the super absorbent that stays in there for a very long time. Blood is a nice, rich growth media, and if it's the right or wrong strain of Staph aureus, they express that toxic shock syndrome toxin that's secreted. It enters the circulation, goes throughout the body, and now you have all those symptoms of a toxic poisoning. You have the fever. You could have the scaling of the skin. Here, that shedding of the skin. You have all sorts of GI symptoms, lots of different symptoms. Not because the bacteria has gone everywhere throughout the body, but because the toxin has. All right. This is not. It doesn't happen only with tampon use and all of that, but it can happen at any time that you're infected with that TSST positive strain. It's just this is where we found out back in the late 70s. This is where we found out about this trait of Staph aureus, because that's when it happened with the production of the super absorbent tampons. History you may not want to know, but there it is. And again, this is a super antigen. Here you will, for food poisoning, you don't treat the patient because the bacteria are dead, they're eating the toxin. But because the bacteria are colonizing the patient, this is where you would treat, especially if there are recurrent events. All right. Scalded skin syndrome, you see this most often in babies. And again, this is where you've got the Staph aureus strains that are producing those two exfoliative toxins. All right. Specifically, these toxins go in and cleave connective proteins that hold the skin together. And so basically, you're just cutting some of those intercellular adhesion molecules, and the skin separates off. And again, the toxins entering the circulation, going everywhere throughout the body. So you see this very extensive shedding of the skin. Okay. Nikolsky sign, just in case you see that in a question. No, I'm not going to put that on an exam, but in the practice questions in the textbook, Nikolsky sign is where you see a little bit of a blister, and then if you put just a little bit of pressure on that, and the skin separates off. And so that's a diagnostic test for the staph and the strep exfoliative diseases, because we're going to see a very similar disease with strep here. All right. But again, it's the toxin that is that's causing the disease. You can look at any of these blisters, any of these skin places, lesions on the patient, and you're not going to find the bacterium, right? Because it's somewhere in the body, but it's not everywhere in the skin. All right. One diseases that may be a little bit more familiar. So our localized infection, whether it's a wound infection, impetigo, and again, we're going to see a parallel disease with strep, but this is where the very surface of the skin, not deep tissue, but the very surface of the skin is infected, okay? And so in that dermal layer, you're getting that infection, little pustules that are going to now, because you're destroying the skin, you're destroying the capillaries, you get some leakage of the serum. And so that's this honey-crusted lesion, and it's because the serum is being released from the damaged tissue. Anybody ever had a sty on your eye? That is Staph aureus. It's a folliculitis, so it's where your eyelash, and it can be on your arm, it can be anywhere, and it has just in the hair follicle, the bacteria are growing, and so you get this folliculitis. Bless you. Now, if lots of those follicles, multiple of those follicles in the area are infected, those individual follicles fuse together to cause, medical term, a furuncle, common language, boils. And now if you have multiple furuncles that form together, that coalesce into a single lesion, that's a carbuncle, okay? Again, in blue, so not anything I will test you on, but here's a really great memory tool because this is something. Furuncle versus carbuncle, what's the difference? Well, remember, it takes 4 people to get into a car. Furuncle are individual little ones, carbuncle bigger ones. All right, and cellulitis. So all of these are very surface. Cellulitis is now going to happen in the deeper tissue. You're getting more now into the subcutaneous. But again, it's just that staph infection in the deeper tissue causing the inflammation. But because you don't have that wound, you're not right there at the skin surface, you don't necessarily have that penetration until maybe you have such a level of inflammation build up that you get that eruption. But again, it's kind of like the volcano erupting from that deeper tissue. Okay. Then we can go down into the deep tissues infections. And so this is where it becomes important that Staph aureus is that facultative anaerobe. Because how is it going to now cause those joint infections or osteomyelitis, infection of the bone? Really common in kids, and especially if with a couple of underlying diseases that are medical conditions that we won't get into. Can cause pneumonia. Okay, so different situations where aspiration, that will happen especially if you pass out and then you aspirate your respiratory secretions, because again, remember, we are infected with a lot of people, over 30% colonized in the nose. And then so here's your heart valve, so here's your different chambers. Here's a valve, but see that white area that is growing right there? That is we call it a vegetation. That's a colony of bacteria that's growing on that heart valve. That is endocarditis. Endocarditis is technically the pathogen, usually bacteria, growing anywhere on the heart tissue, but overwhelmingly, most frequently, that's occurring on the valves, okay? So bacteria is in the blood. You've got some damage to your valve. That damage gives it kind of a rough surface, a good place for the bacteria to hang on to, especially Staph aureus, they've got fibrinogen, and now they become anchored there. All right. And so MRSA, remember methicillin, because I think we talked about that. Methicillin is a very shelf-stable, so that's what it's used in a lot of diagnostic labs, but it's a member of the beta-lactam family. It's a first cousin of penicillin, if the name gives it away, so it has that beta-lactam ring. And that was used in testing to determine, and that's how we determined that Staph aureus had acquired that resistance gene. The resistance gene is encoded by MEK-A. It was picked up on a plasmid, and it encodes a penicillin binding protein that looks a lot like the transpeptidase. So that's how it got incorporated in the mechanism of action. It looks like a transpeptidase in that it binds to the peptidoglycan, it binds to the binding site, but it doesn't mediate it doesn't form that transpeptidase bond that the normal, or the bacterial proteins, not ours, but the bacterial proteins, the normal transpeptidases that perform that cross-linking, this gene does not do that. But it can now bind the antibiotic, the methicillin, the penicillin, at a very low affinity, but not high enough that it's going to kill the bacteria. So there's the mechanism of action. It prevents binding of the drug, but because this is now an exogenous gene, the bacterium is making a lot of it, and so now the penicillin, the methicillin, whatever cellin cannot, whatever beta-lactam cannot work very well, and now you've got your drug resistance. Big risk factor there is healthcare exposure. This is, really sick people in the hospital a long time. This is where it circulates, but it's not exclusive. We do have community-acquired strains of MRSA as well. All right, ready to move on real quickly to Staph epi. So, much less virulent than Staph aureus. That's why it can be part of our microbiome on the skin. Why is it less virulent? Because it has a lot less virulence factors. Causes less severe disease when it does get into the body. Less severe disease, more indolent. Indolent means a slower disease process, less aggressive symptoms. So instead of having a fever of 103, it'll be maybe 100. That low-grade fever, achy, all of this, but it's just not super aggressive. You're like, oh yeah, I can deal with this. No, you can't. Okay. And because it is less virulent and because it's part of the flora, this is now that opportunistic disease as we talked about earlier. When the staph get from the skin into the deeper tissue, and then it overcomes the host defenses and causes disease. Okay. Again, that surgical infection, prosthetic heart valve. Here is a prosthetic knee that has been removed, or here when it's in place. See that little cloudy area right above the arrow right there? That is your vegetation. That, yes, here's the hardware of that knee, but that little cloudy, you can actually see it starting to erode the bone there, that little dip. Right? That's the infection. All of this tissue here, and it's not just tissue from the patient, this is where the staph epi has been stained, because when that joint was implanted, either it got contaminated during the implantation, or it may have become contaminated with some staph epi on it from the manufacturer, not unheard of. Same thing with a heart valve. Here's the endocarditis, so here's good healthy tissue, all of this yellow deposition. These are the vegetations, that's the bacteria growing on that heart valve. And the reason it can do that is because, again, remember this is a staph, they form biofilm really well, so the biofilm helps that adhesion. All right. Staph saprophyticus, real quickly, I'll give you this one. This will not be on your exam, but it is, in the learning objective, it is 1-3, really medically important. So this is almost where we think about this really exclusively is urinary tract infection, also called cystitis, and especially in younger sexually active women, right? Not that it's only women that are colonized with this, but remember it's in the genitourinary. Having a nickname for this disease is honeymoon cystitis. So, you'll learn about in medical school if you choose that path. But this is where you have lots of very frequent sexual activity, and now it's just kind of, thinking colonization of the genitourinary tract. The bacteria get implanted in a deeper tissue and not just in the genitourinary, but much deeper into the bladder because of all the contact. And now there's enough bacteria there that the host defenses can't deal with it, and you end up with a staph infection, with the staph saprophyticus infection, a urinary tract infection. Okay, so when we go through the testing for this, again, we've talked about the gram stain, then you do your Cadlase test. Here's the algorithm. You've got a Cadlase positive, you know it's staph. You do your coagulase test. If it's coagulase positive, you know you've got Staph aureus. If it's coagulase negative, then you're going to think, I'll come back to these points, but if it's coagulase negative, then you're going to say, all right, is it Staph epi or is it Staph saprophyticus? That's when, if you're following over here, here's a coagulase negative, that's when you then do a novibacin test. Novibacin is an antibiotic. So you'll streak that plate. We call it streaking for a lawn. Think about a lawn of grass. You streak the plate with your bacterial culture, and then you drop that disc, which would have novibacin on it. Okay. See the little halo of no growth around that paper disc? That's your susceptibility. That's no bacterial growth. If you would see that pattern, you say that it is sensitive to the antibiotic because the antibiotic is inhibiting the growth immediately around that disc, right? Okay. So if we're talking about staph and novibacin, we know that this would be staph epidermidis. If it were staph saprophyticus, which is novibacin resistant, then your bacterial growth, the lawn would be all the way up, and even if you lifted off the disc, it would be under that paper disc. That's your resistance. All right. Everybody understand how to interpret these tests now? Yeah, okay. There it is. Okay, so if we go back to thinking about these points. So Staph aureus, most strains, but not all, are beta hemolytic. Remember, beta hemolysis is where you have complete hemolysis when you streak that when you streak that blood agar plate. All right, this is your beta hemolysis. Your other strains of staph or your other species of staph are going to be gamma hemolytic, so no hemolysis, all right? So you never want to go just based on your hemolysis, but again, that's another good clue that this could be Staph aureus. A conclusive step would be putting it on mannitol salt agar. Only staph are going to grow on the plate, but here's your coagulase negative growing. Let's see all of those nice white lines, cream-colored lines. That's your colony growth. So it can grow on there. If this were any other bacteria, you might see the kind of the pattern of the streak, but there would not be that cream-colored bacterial growth. All right. So that would be an inhibition. But here is, so that is your differential, or your selective media. Differential is where we're getting growth of both, but now you've got the color change. This would be the color of the negative, of the standard mannitol before you streaked it, but it's turned yellow. Why is it turned yellow? This is Staph aureus. It can now ferment that mannitol sugar. What's an end product of fermentation? All of those acidic metabolites. What is an acidic metabolite? It's going to drop the pH. And now, depending on which pH indicator is in the media, there's going to be a color change. For mannitol salt, for a lot of the media, it's going to go from a pink color to a yellow. That's a change in the pH, an acidic pH. For some of them, it will be a different change, but we'll talk about that when we talk about that media, when we talk about the gram negatives next week. All right, everybody got that? So mannitol salt is highly selective and differential for staph, and especially Staph aureus. All right, we will start talking about the strep and then finish them in the next hour. All right, so your streptococcus, again, for the whole genus, they're growing in chains. So they're gram-positive cocci, but instead of growing in clusters, they're growing in chains. They can be very long chains, or they can be very short chains, as in a chain of 2, as we'll see with some of the organisms. Okay, but you've got chains. They're catalase negative. Remember, you come up with you get a gram-positive coccus. What's your very first test? You're going to do the catalase. Catalase positive, it's staph. Catalase negative, it's strep. And then you continue on. Okay. We're colonized with a lot of the different strep at various parts of our body. So there's a major virulence factor, the fact that it's already there. Some of them, yes, there's going to be a clear transmission before you have disease. Some of them, it's just that it's going from that colonization, especially in the upper airway, to now a different tissue where it's going to cause the disease. All right. So how do we organize these strep? There is this thing called Lancefield grouping. And does this look a little bit like your blood testing that we talked about? Your blood, you determine your blood test. It's the exact same principle. All right, so this is based on a lot of the strep have a polysaccharide capsule. And so those polysaccharides, yes, it's all a polysaccharide capsule, but there are different polysaccharides in each of those. And now different polysaccharides means different antigens, right? Different antigenicity. So the Lancefield grouping is based on the ability of these bacteria, or the immune response, to make antibodies to those different polysaccharides. That gives us now antibodies that we can serotype the bacterium. Okay, sero because we're using antibodies. Loose use of the term, but that's what we call it, right? So this is through an agglutination test, because in each of these little vials, there is a latex particle that those antibodies are linked. So in the red tube, it's going to be an anti-group A antibody. In different groups, because for the Lancefield test, for the Lancefield grouping, you have groups A through G. Each of those letters is a different antibody specificity. So that's where you have these different antibodies. You put some of the bacteria in your little dot. You put a drop of the antibody solution with the antibodies linked to those latex particles. And then if the antibody binds to the bacteria, you get that agglutination. Just like you do with your blood typing if you've got the anti-A, B, or O. Okay, just 1 second. And so that's how we determine which serotype it is. Yes?
Is there not an E?
No, there's not. There is, but it's not medically relevant. So as far as what we're thinking about, no E. These are the medically relevant, okay? And yes, there are some that do not have a Lancefield grouping because we can't seem to make an antibody that works in this assay for those. One of the major ones, all right? And like strep pneumoniae, there it is, all right? Okay, so when we talk about some of these organisms, I'm going to give you the Lancefield grouping, all right? But we're really going to think about them by their specific names, all right? And so that's one way that you will start your organization. And then the next test, you know you've got catalase negative, then you're going to streak that blood agar plate and you're going to look for your pattern of hemolysis. Is it beta, alpha, or gamma hemolytic? All right. Again, there's another thing that, just like I said earlier, it's all about the slide number, the supplemental slide number. That's the only thing that star means, okay? So, pathogenesis, as I mentioned, one of the major pathogenic virulence factors is the fact that they establish that the streptococci establish those commensal relationships. We're already colonized, so we don't have to have a separate transmission event, even though for some of them there will be a transmission. That staph pneumoniae, somebody coughs or sneezes and you inhale those respiratory droplets, right? Other pathogenic features, general rule of thumb, beta hemolytic are more pathogenic than either the alpha or gamma. Why is that? Why is there beta hemolysis? Because that bacterium is secreting a hemolysin. So they're secreting an enzyme that are going to penetrate, going to lyse that red blood cell. Well, those hemolysin, we call them hemolysins because we can easily see lysis of a red cell. But there's not that high specificity. They don't lyse just the red cells, they can lyse your other cells, like your neutrophils, as we'll see with one, okay? And so now you've got that pore-forming activity, which is going to cause more tissue damage, right? They can elicit inflammation, invade the mucous membrane. So lots of the enzymes that they'll make are now going to cleave the mucus, and so now why is that an advantage? It gets you the bacterium right down there at that epithelial layer that they can bind and now establish that infection. And then there's lots of other factors that are going to be specific to the individual organisms. Okay? All right. Excuse me. This will be a good place to stop. We'll go ahead and take a 8-minute break, start back at 9 o'clock. We'll finish off talking about